Hi everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. In this session, we will be discussing about 7 mistakes which we need to avoid during DNA isolation, especially in the biotech and molecular biology lab. So let's get started. Number 1 is using dirty equipments and instruments. Always ensure your equipments and atmosphere is clean and free from contaminants. Cleanliness in the laboratory is the key. The second mistake is skipping the sample homogenization. Don't forget to thoroughly homogenize your sample. It is going to help you ensuring uniform DNA extraction. Incorrect buffer selection. It is also essential to choose the right buffer for your DNA isolation method. It can significantly impact your result. The next one is insufficient lysing time. Give your sample enough time to lyse properly. Rushing these can lead to low DNA yield. The next one in the list is skipping the precipitation step. As you know, precipitation steps are crucial for DNA purification, so don't skip them to avoid the impurities. The next one is neglecting the contamination control. It is also important to be vigilant about the contamination control. Prevent DNA contamination from the external sources. And lastly, the poor storage practices. Store your isolated DNA properly. You might be needing them in your future experiment. Follow the recommended protocol to maintain its integrity, and by avoiding these common mistakes, you will achieve high quality DNA for your experiment. Thank you for watching.